Okay, so we're going to be starting the lecture on chromosomal anomalies. So this is going to be the series of lectures that we're going to be going through. It'll be a little bit more, it's more not so much problem solving as we've done, not so many steps as we've done in the past 4 or 5 lectures. But this is just more informational, just more memorization and information to know. And this will build upon the last 2 or 3 lectures that we have remaining. So a chromosome anomaly or a chromosome abnormality or an aberration is essentially a missing, it could be an extra or an irregular part of chromosomal DNA. And it could be from an atypical number of chromosomes, or it could be a type of a structural abnormality in one or multiple chromosomes for that matter. Chromosome anomalies, usually they happen when there is an error in cell division after meiosis or mitosis. Although there are many types of chromosome anomalies, they can be organized into 2 basic groups. So they can be either numerical or they could be structural abnormalities. So we're going to be looking at both of these in a little bit more detail, and we'll be looking at examples of them as well. Before we move forward, we should have a good understanding of what a karyotype is. So a karyotype refers to the full set of chromosomes for any given organism, which we can compare to let's just say a normal karyotype for that specific species via genetic testing. So again, if for example, this is the normal, okay, and by normal we're saying that there are not any abnormalities or any defects that are present, major defects. So if you look over here at this, essentially, if you count the number of chromosomes, there will be 46 chromosomes. So this is the first pair, second pair, third, fourth, fifth, sixth, seventh, eighth, ninth, etc., etc., etc. pairs of chromosomes. So essentially, we have a total 23 pairs of chromosomes. 23 pairs of chromosomes, 46 total chromosomes. Now, this set over here, the 23rd pair, this is the sex chromosomes. So this will dictate whether the organism is male or if the organism is female. Okay? The rest of these chromosomes over here, all the others, the other 22 are what's called the autosomal chromosomes. Okay, so autosomal chromosomes and the sex chromosomes, the sex pairs. So we have 22 pairs of autosomal chromosomes, and then we have 1 pair of the sex chromosome. This is chromosome pair 23. So starting off with numerical disorders, numerical disorders, we use the term aneuploidy, okay, aneuploidy to describe that. And in aneuploidy, we're saying that there's an abnormal number of chromosomes. So this happens when an individual is either missing a chromosome or they have an extra chromosome. So again, remember, they're either missing a chromosome from a pair, and again, that if they're missing, that would be called monosomy. And if there is extra chromosomes in the pair, so if it's more than 2, then we call that polysomy. So monosomy, there's just 1 chromosome in the pair, and polysomy, there's more than 2 chromosomes that's supposed to be there in the pair. So we're going to be looking at examples of all of these in the next few slides. Numerical aberrations represent a significant portion of chromosomal changes that are found in humans. Now, we can say that they come in 2 types. The first one in constitutive. Constitutive, we're saying that this is going to be present in every cell of the body, right? And this takes place during meiosis. Okay, this will take place during meiosis. In mosaic, we're saying that it's not present in all cells, it's present in some cells. And this takes place during mitosis. All right? So remember, for this one, what we're saying is that every single cell in the body is going to have either 3 pairs or 2 pairs, or not 3 pairs, I'm sorry. There's going to be 3 chromosomes instead of 2. All right? Or there may only be 1 instead of 2. Or there could be more. So this is what constitutive means, okay? It's for every single cell of the body. In mosaic, okay? Mosaic, think about it, guys. It's a pattern, right? Sometimes you have more, others you don't. So in mosaic, it's not every cell. It's some cells that are affected, right? So they both of them, they represent a significant cause, a big cause of pregnancy losses. So again, spontaneous abortions, miscarriages, as well as abnormalities that we see in live births. So of the first of these numerical aberrations, we are looking at monosomy. And remember, in this one, we're saying that there is 1 chromosome that's missing. So when you look over here at this karyotype, karyotype is for Turner syndrome. And you see over here, when you go to the 23rd pair of chromosome, there is just 1 chromosome present. Okay, so there should be 2, right? This is a female. So there should be an XX. However, there's just 1 X. And again, this is for Turner syndrome. And Turner syndrome could be mosaic, meaning that not all cells may be affected by this. So Turner syndrome is associated with the underdeveloped ovaries, a short body stature, and we only find Turner syndrome in females. So it's also known as 45XO Turner syndrome. So meaning again, there's only a total of 45 chromosomes, and there's 1 X, and there's a missing chromosome. Okay, so 45XO syndrome is also known as Turner syndrome. Other things that we see with Turner syndrome, aside from what I said before, the short stature, the underdeveloped ovaries, is that there could be, when you look at the neck, there's a broad neck over there, a bulky neck, a broad, again, if you go back here, there's a broad chest that's present as well. You can see some scoliosis over here that's present as well. They lack secondary sexual characteristics as well. Aside from that, mental retardation is not evident, fortunately. You may see a low hairline that's present. There's these certain characteristic features that you have. There could be folds within the skin that you may see as well on certain parts. It could be on the face. There's a cardiovascular problem as well that results in. One of the things that we see is that there is a constriction of the aorta that you see. So again, this causes cardiac complications. So there's a handful of phenotypic characteristics that may show up in Turner syndrome. So be sure you go over this and you're aware of what all these signs and what these symptoms are. Moving along, so sometimes we see a deficiency in a chromosome. Other times you see extra chromosomes present. And one of these disorders that's rather common, as a matter of fact, 1 in 700 births would result in Down syndrome. So this is how common it is. And what we see over here is we see an extra chromosome on the 21st pair of chromosomes. So on the 21st pair of chromosomes, there are 3 chromosomes present instead of 2. And about 2/3 of Down syndrome cases result in a spontaneous abortion. And usually the extra chromosome comes from the mother most of the time. We do see a correlation with the maternal age. So for example, a woman that's 25 years old has perhaps 1 in 2,000 chance of having a baby with Down syndrome. Now when you compare that to a woman that's 10 years older, now we're talking about a 35-year-old woman, that risk increases to 1 in 350. So 1 in 2,000 for a 25-year-old to 1 in 350 in a 35-year-old. Now, by the time a woman is in her 40s, Now that risk has gone up even more. So a 40-year-old has a 1 in 100 chance of giving birth to a child with Down syndrome. Raise that up to a woman who is 49 years old. Now that risk is 1 in 10. So these numbers, you can clearly see that there is a correlation with the maternal age. The higher the maternal age is, the higher the probability that a child with Down syndrome will be born. When you look at a person with Down syndrome, symptoms, they include an upward slant to the eye, as you can see. Aside from that, small ears are present. Again, they fold over at the you can see it over here. They kind of fold over at the top. There's a small flattened nose, small mouths, and their mouth also gives the appearance of the tongue to be larger. There's a very profound short neck that's present, and also when you look at the hands, you'll see that the hands are short. Okay, there's small hands and short fingers that are present. Now, there is a certain degree of mental retardation, and this will range anywhere from being mild to severe. In most cases of mental retardation, it's usually mild to moderate, though, right? So, while the gamut ranges from mild to severe, the vast majority is mild to moderate, fortunately. Trisomy 18. So when you look at this karyotype over here, again, on the 18th pair of chromosomes, there is an extra chromosome present over here, okay? So again, this is where the term trisomy comes. Now, one other thing I wanted to mention, let me go back. Down syndrome is also called, can you guess it? Trisomy 21, okay? So there's 3 chromosomes on the 21st position. Therefore, it's called trisomy 21. Now we're looking at trisomy 18. On the 18th position, there are 18 chromosomes instead of the pair that should be present. So, trisomy 18, it's also known as Edwards syndrome, or they also call it 47XY plus 18. So there's a total of 47 chromosomes, all right? And in this, 47XX in this case, because there's 2, this is a female, so 47XS plus 18. The plus would mean there's an extra chromosome on the 18th, in the 18th pair, that's where there should be 2. Now, what's the risk for this? Fortunately, it's much less than Down syndrome. It's 1 in 5,000 on average. Girls are affected far more than boys are. Okay, so again, usually this results in, you tend to see this more in females. Any woman can have a child with trisomy 18, but again, that risk does increase with age, as we saw in trisomy 21. So when you look over here, there's a low birth weight of a child born with trisomy 18. There's a small mouth, a small jaw that's present, an abnormal shaped head, again, small head. Fingers are going to be, perhaps you can see they're overlapping, so you have overlapping fingers. We'll see a clenched fist also that's present. They also have these what they call rocker bottom feet. When you look at the ears as well, there are low set ears that are present. And, probably the worst of this is going to be there are defects in the heart and other organs. Now, we're going to be moving on to structural abnormalities, okay? So we talked about abnormalities in terms of numbers. Now we're looking at structures. So structural aberrations, again, these are changes that take place in the chromosome structure. And there could be structural chromosome rearrangements. And again, these rearrangements, or these changes occur and are applied, to the physical integrity, to the physical structure of that chromosome. And again, these structural changes, they could result in birth defects, mental retardation. It could increase the risk of infertility. They can also lead to spontaneous abortion. Again, this means pregnancy loss. So again, these are, they happen most of the time due to chromosomal breakage, or it could be unequal crossing over. And again, remember, we're talking about events that are taking place in meiosis. So this results in deletions in ring chromosomes, and duplications, translocations, inversions, and insertions. So we're going to be talking about what all these mean, okay, as we go through through this lecture. So when you come over here, again, this is a good normal chromosome. All right, and what are we looking at over here? So over here, we're seeing that, again, this chromatid, right? We have sister chromatids that make up the chromosome over here. So when you look over here, this is a short arm, which is called the P arm, and this is a long arm called the Q arm. Okay. By the way, the P stands for petite, and the Q stands for Kie. So now, the other thing that you notice is that, there's all these lines and stuff, right? Now, remember, what is all this? This is genetic information. This is DNA. This is a super condensed chromatin, essentially. That's what this is. So, again, what they do is, again, for the purpose of locating genes, they end up dividing this, okay, or organizing this. So, again, it's called chromosome organization. So, what ends up happening is this. They end up naming a specific part over here. Right. So, for example, they'd name this part. What is this line over here? So this would be, they would say that this is not located, or this is found on the P arm, okay, in the second region of the second band, in the second subband. All right. So this is how it works. So each chromosome is divided into regions, and then it gets labeled. Again, is it P or is it Q? So if you're looking over here, it would say it's the P arm or the Q arm. And then it's labeled into the region. Which region is it? And we're looking at region 1 or region 2. After that, we're looking at which band are we looking at. All right? After the band, then we're saying what's the subband. So this is how they end up dividing it. So again, they might read this as, for example, let's see. All right, so it's a P arm 111. Okay, that would be here. P arm 111. That's how this ends up getting read. Or you can say it's Q arm 233. All right, so this is how it would be labeled. What's the other thing? All right, so look, you see the centromere over here, and then these are the telomeres at the ends, right? So telomeres here and there, and then we have the centromere in the middle, in the centromere. All right. So the first structural abnormality that we're going to be looking at is deletions. Okay, a deletion. So deletions, again, what ends up happening is a portion of that chromosome is either missing or ends up getting deleted. It gets removed. So known disorders in humans, an example of this would be, for example, Wolff-Hirschhorn syndrome. Okay, Wolff-Hirschhorn syndrome. We'll look at this in the next slide. And again, this is caused by a partial deletion of the short arm of chromosome number 4. There's also Jacobson syndrome. And in Jacobson syndrome, it's also called, it's the terminal 11q deletion disorder. So again, when you look over here, essentially what ends up happening is you end up losing part of the chromosome, and then it ends up getting lost. So this is that normal chromosome. Then you have a break. This part breaks off, and then this is lost. This is all that's left. Okay? So a terminal deletion. This is where a terminal deletion would be. An interstitial deletion would be that, again, you have this chromosome here. This part breaks off. These two ends come back, connect together, but this part ends up getting lost. Okay? So terminal deletion versus an interstitial deletion. So the prevalence of Wolff-Hirschhorn syndrome is estimated to be about 1 in 50,000 births. Now, almost everyone with this disorder has this distinctive facial features that include a broad, a flat nasal bridge that you can kind of see over here and there. And again, if you look at the forehead, we have a high forehead that's present as well. Aside from that, there is also the eyes. We have wide eyes that are present. Other characteristics include there's a shorter distance between the nose and the upper lip that you can see over here. Aside from that, there's a downturned, again you can see over here, a downturned mouth that's present. There's a small chin, and again, the ears they're underformed, so you have the poorly developed ears, and they have small holes also. Now, in these individuals, you can expect delayed growth and development. So motor skills such as sitting, standing, and walking, they're significantly delayed. Microcephaly, or a small head, may also be present. And most children and adults with this disorder, they also have a short stature. Next, we look at duplications. And in a duplication, a part of the chromosome is duplicated. It's copied, and this results in extra genetic material. Known human disorders of duplication, there's something called Charcot-Marie-Tooth disease. We'll look at that. But again, when you look over here, this is when you look over here, this is the deletion that takes place. So if you're looking at this chromosome over here, what ends up happening is we end up getting this part of this chromosome ends up getting doubled over here. So look at this band over here. Now this is nearly twice as large. So again, this is a duplication would be. So in other cases, versus a deletion, if this is a normal chromosome and this part gets removed, now we have a deletion. So again, the previous one, we looked at deletion, where we lost genetic information, which is this. Now we're looking at duplication. In duplication, again, what are we saying? We have some genetic information, extra genetic information, okay, that we have. So maybe it's doubled, maybe it's tripled. So this is essentially what duplications are. So in Charcot-Marie-Tooth disease, or CMT, this is a hereditary motor and sensory neuropathy. It involves the PNS, the peripheral nervous system, and this is characterized by progressive loss in muscle tissue. Along with that, you lose touch sensation, and this takes place across various parts of your body. This disease, mostly we see this as being an inherited neurological disorder, and it affects about 1 in 2,500 people. So again, when you look over here, here's some common characteristics of this. You look at the hands that are affected; they're somewhat clubbed and bent, as well as the foot, where they have this excessive arc over here that forms, and again, abnormal structure. So again, these individuals, they have problems with walking and grasping objects. Next, we move on to translocations. Now, translocations involve breaks in 2 different chromosomes with an exchange of segments. In humans, there are 2 major types of translocations. Okay, the first one is a reciprocal translation, and the second is what's called a Robertsonian translocation. So in the first one, in a reciprocal translocation, this is where there's no visual loss of chromatin, right? In a Robertsonian translocation, this is where the long arm of 2 acrocentric chromosomes are connected with the loss of 2 short arms. Okay, so keep this in mind, and we're going to see a picture of this in the next slide. So again, remember, the 2 long arms of an acrocentric chromosome come together, and the 2 short arms are gone, are lost. So here we go. Now, what do we see over here? This is a acrocentric chromosome. So now, again, we're looking at a Robertsonian translocation. So what ends up happening is this part breaks off. All right, so now you got the break that takes place. So this part, and this is another chromosome over here. What ends up happening? This part breaks off. So again, the short arms both fall off and get lost, and the long arms come and connect together. So this is the new chromosome over here, right? So here, this is the normal chromosome over here, chromatid. This is the short arm, this is the long arm. Remember, we're talking about acrocentric. What is this over here? That's a centromere. So this part breaks off and it gets lost. And on this other chromosome over here, this part breaks off and gets lost. Now we're just left with this part for this chromosome and this part for this chromosome, okay, over here. So these two ends come together and connect, right? That's it. That's a Robertsonian translocation. In a reciprocal translocation, what's happening? Oh, well, here, we got this one normal chromosome here. This part breaks off. Now, this part breaks off, it goes to, so remember, this is a short arm, this is a long arm. This is one chromosome here, and this is another chromosome here. This chromosome here, again, long arm, short arm. So in this one here, what ends up happening? Again, you end up getting a break over here. So this break goes, this is a long arm, right, over here. Short arm, long arm. So this long arm will go and connect over here, and the short arm that broke off will come and connect to the other end. So, I'm sorry, yeah. This is what will come connect to this one over here. So this that you see over here is from this chromosome, and this that you see over here is from this chromosome. So essentially, you're having an exchange of the arms that are taking place, right? That's all that's happening. So what do we see over here? For this one, we said what? We ended up losing the arms, right? That's in Robertsonian. In the reciprocal translocation, we don't lose the arms. The arms just get swapped out between the two chromosomes. Okay, that's all that happens. The short arm goes and connects with the loss of the loss that took place of the other chromosome, and this chromosome that lost it will come and replace the part that broke off from the other chromosome. That's it. It's reciprocal. I lose, this one loses, he gets another one. This guy loses, he gets the other one as well. So they both exchange that. Next, we look at inversions. And inversions are formed by two breaks in the same chromosome with the exchange of the two ends. So there's a couple of different types. There's a pericentric inversion and there's a paracentric inversion. Okay? Pericentric and paracentric inversion. So for a pericentric inversion, this is where one break occurs in each arm of the chromosome and the centromere is included in the inverted segment. Okay? Sounds pretty simple. So if you look over here, here's a pericentric inversion. Here's the centromere is right over here, right? This part is the centromere. You get a break over here, you get a break over here, and this part just flips around. The yellow part now goes down, and the red part or the maroon part goes up. That's all a pericentric inversion is, right? Pericentric inversions, they involve the, in this case you're looking at over here, what's involved at the centromere. Okay, so the centromere is included. Centromere is involved. These are breaks that involve the centromere. Now we look at the opposite. Okay, a pericentric inversion. So over here we find again the chromosome ends up getting formed when we end up having breaks that occur. So in this case, again, we're looking at this red region here and this green part over here. So this part breaks off. You get a break over here and a break over here. Now what ends up happening? It gets flipped upside down. The red goes to the bottom, the green part goes to the top. Okay, that's it. Pericentric. Right? It happens away from, somewhere around the chromosome. It doesn't happen in the middle. Pericentric, it happens with the centromere. This does not happen with the centromere. Again, inversion. Inversion is you're flipping something upside down. Okay? So again, both breaks, they occur on the same arm. Okay, so the centromere, it's not included in the pericentric one. That's it. That is the difference between a pericentric and a pericentric and a pericentric inversion. Next, we look at insertions. And in an insertion, we're looking at a part of the chromosome that's been deleted from its normal place and inserted into another chromosome. So let's look over here. We got over here chromosome number 4, right? Now, this part is going to end up being inserted into chromosome number 20. It's going to go over there. It's going to be inserted. So what ends up happening is we lose material from one chromosome, and then we add that to another chromosome, right? So what ends up happening? One chromosome ends up losing. So if you compare this to this, now chromosome 4 is shorter. And if you look at chromosome before and you look at chromosome after, it's the other chromosome gets bigger, gets larger. Okay, so a couple examples of this of insertions. One of them is Huntington's disease, and the other is Fragile X syndrome. When you look over here, this diagram over here in green is for Huntington's, and this is for Fragile X syndrome. So what do we see? So for Huntington's, again, there's skeletal muscle. It affects multiple tissues, as you can see. So we have digestive system that's affected. Okay, decreased insulin production, altered gene transcription in the islets of Langerhans. There's decreased levels of T3 in terms of blood. We have mitochondrial dysfunction, increased caspase activity, increased inflammatory markers. So again, they have a high interleukin 6. Heart, okay, the heart rate, it slows down. There's decreased heart rate variability. There's also altered cerebral blood flow, blood going to the brain. There's changes in the muscle tissue as well. So there's mitochondrial dysfunction. They don't produce as much ATP. There's bone disorders as well. Okay, osteoporosis, genital system in men, decreased levels of testosterone. Therefore, the number of spermatids decreases. Therefore, there's a decrease in the number of seminiferous tubules. So again, infertility. When you go to Fragile X syndrome, again, there is behavioral aspect that changes. Now, there is a degree of intellectual disability that these individuals may have. When you start looking at their physical features, the ears, again, they have large protruding ears. There's a long face. The palate will have a high arc to it. They also have hyperextendable fingers, right? So again, you can say they're double joint. So as in the case of the thumb, a double jointed thumb, as well as fingers that are, again, that extend beyond the normal range of that of most people. They'll have flat feet. Their skin will be very soft. So again, these are some of the things that we see in Fragile X syndrome. Next, we move on to rings. So in rings, what ends up happening is a portion of the chromosome breaks off, and then that broken part ends up forming a circle, or, again, the term that you see over here, a ring. So this can happen with or without loss of genetic material. So one of the conditions that we see is something it's a condition that's called ring chromosome 14 syndrome, right? So this condition, it's characterized by seizures and intellectual disability. So again, the recurrent seizures are recurrent. So again, they're epileptic, and this develops in infancy or in early childhood. So when you end up looking over here, what ends up happening is the telomeres, they end up falling off. Okay, they end up getting deleted. So this is this genetic material that deletes. So so remember what I should have said is that this is the 5' caps. So remember this 5' caps is this TTA GGG, TTA GGG, TTA G. This sequence ends up repeating maybe about 2,500 times. And again, one of the things that this does is it prevents the ends of these chromosomes from sticking onto other chromosomes, or in this case, from forming a ring. So once the 5' caps, the telomeres fall off, then this chromosome curls up and turns into a circle or a ring. An isochromosome is an abnormal metacentric chromosome that's formed by the duplication of one arm of a normal chromosome along with the deletion of the other arm. So both arms of the metacentric chromosome are genetically identical. The most common isochromosome ends up being the X chromosome. Now, the acrocentric autosomal chromosomes that are common candidates for these isochromosome formations, they typically occur on chromosomes 13, 14, 15, 21, and 22. So, chromosomes that contain smaller arms are more likely to become isochromosomes because the loss of genetic material in those arms could be tolerated rather well. So in individuals with the isochromosome, the appearance of the face and the digits, again with fingers, they tend to be smaller to those observed in another type of syndrome that's called Pallister-Killian syndrome. We also see cleft palate, there's diaphragmatic hernia, and also, unfortunately, there's a neonatal death also that ends up resulting. So again, when you look over here, let's see what's going on. So again, we have normal chromosomes here. This is the P arms, and these are the Q arms over here. So what ends up happening now? If you look over here, these over here, this short arm is going to end up going to the other one. Okay, so they get switched around. So this one will end up going to the other chromosome, and this long arm will come and connect with this one over here. So now we have two Q arms on this one chromosome, and the other chromosome is just these two P arms. So this is what isochromosomes are. All right, so let's talk about inheritance. Most chromosome abnormalities, they occur as an accident in the egg or sperm, and therefore the anomaly that it presents, it tends to occur in every cell of the body. Now, some anomalies, they can happen after conception, and this is what ends up resulting in mosaicism. So these chromosome anomalies, they could be inherited from one parent, or it could be from both parents. Now, the other thing is that if it's not inherited from a parent, then it could be a mutation. So, again, this is why chromosome studies are often performed on parents when a child is found to have, some form of an anomaly. So if the parent does not have this the abnormality, that means it wasn't initially inherited. However, it may have been transmitted to, again, subsequent generations. So, again, these chromosome abnormalities, they usually occur when there's an error during cell division. So that's it for this lecture.